I see now that there's less people in the waiting room, so I suggest we actually give it a start. And it's really my pleasure to welcome Hans Bernstein to this week's Planetary Biology Lecture. And maybe a little bit of background. Hans, I know you will cover some of it, but Hans, he did biological and chemical engineering at Montana State University, where he received his bachelor from and also his PhD. He then built his independent group at the Pacific Northwest National Laboratory in collaboration with the Washington State University, where he stayed for 5 years until in 2018, he moved to Norway, to Tromsø, where there's already the midnight sun now, as we just learned, where he is an Associate Professor of Biosciences, Fisheries and Economics at the Arctic University of Norway. And he leads the Microalgae and Microbiome Research Group. He's also associated with the Arctic Center for Sustainable Energy as the manager of the greenhouse gas research portfolio. And we had the opportunity to talk a little bit together before, and I'm really fascinated by the connection of fundamental research and also linking that to a very applied questions, being at the intersection of biotechnology, but also microbial ecology, and applying that to the fields of sustainable bioprocess engineering, marine microbiome, synthetic biology, and CO2 capture. And today, Hans will talk about Hot Lake, a story of salty biofilms, microbial activity gradients, and succession dynamics. Hans, thanks again very much for accepting the invitation and for being with us today, and the floor is all yours.
Thank you, Michael. I'm going to share my screen now. Hope this goes well. It's a pleasure to get to be here with your Planetary Biology Lecture Series, and I'm broadcasting to you today from my office in Tromsø, and it's very sunny, which means seagulls are having a party on my windowsill, so I apologize for that. It's not weird noises coming from me. So today I'm going to talk a little bit about myself, not because I'm a narcissist necessarily, but just because I find that I always like to hear a little bit about the speaker. I'm going to talk a little bit about my research group, the M2RG, and then I'm going to talk about science, which is my favorite thing to talk about. I've had great conversations with people today, and then just give a little bit of a prelude of what's coming next. So I am, even though I live in northern Norway, I am not Norwegian. I'm from the US, and I grew up in the state of Montana, which is, it's the size of Germany, but it has only 1 million people. And it has a pretty unique education environment at Montana State University. It has a great engineering school, but all this focus on microbiology, specifically like geochemistry integrated with environmental microbiology. So I was really lucky to do a PhD there and learn this interdisciplinary aspect of microbiology and engineering that really set the trajectory for the rest of my career. And then when you get a PhD, you're lucky if you get a job. And I was lucky. I started as a postdoc under a fellowship program at the Pacific Northwest National Laboratory, and then was promoted to start my first research group there. So that's where I cut my teeth as a PI and figured out, well, how do I run my own research projects? And so this is a really big multidisciplinary environment, fast-paced, well-funded science. It's operated for the US Department of Energy. So there's a lot going on there. The problem that I had was not the science. I had great programs in managing both synthetic biology and environmental microbiomes. And I'll talk a little bit about some of the research I did while I was there today. The problem I had was that the US Department of Energy is part of the executive branch in the US, which came under new management. While I was there, we gained a new president. His name was Donald Trump. And pretty soon I found myself being pushed more and more into projects I didn't want to be a part of, different aspects of national security. and whatnot. So I looked for a new job, I looked for a new adventure, and I found myself here in northern Norway, which I had never been to before. But now I live here, and I've been here for 3 years. I live in Tromsø, which is a medium-sized city in the Arctic. It's literally a city on an island or a couple of islands that connect to each other and connect to the mainland. The university it's not giant and it's not too small, but it's relatively new, and there's this idea of we can do anything and we have access to this marine ecosystem in the Arctic, a gateway to everything. And it's a really exciting environment. And it's also a great place for a work-life balance. I love the mountains, I love the snow, I really like to fly fish, I like to spend time with my kids. So things are about as perfect as they can be here for me. So 2 years ago, I initiated a new research group here, the Microalgae and Microbiomes Research Group. And we have 3 pillars of research, or 3 focus areas. And for me, the really exciting science happens at the interconnection between these pillars. So I'd say the majority of our research, or maybe 50% of it right now, is funded to do microalgae research or applied microalgae research for the purpose of carbon capture and utilization. And then we have a pretty large focus on marine microbiomes, like understanding how microbial ecosystems interact both in the subarctic ecosystem we live in here, but also we spend a lot of time up north on icebreakers looking at what's living under the ice or on the ice shelves. And we're pretty interested in how microbes interact, what drives interactions, and how do these interactions structure community dynamics and biogeochemical cycles, and obviously the carbon cycle, so how that can be translated to different biotechnologies. And then perhaps the fastest growing area of our program here, and sort of what I brought with me when I moved here, is biodesign and synthetic biology. And the idea here is learn to program and control microbes. But what we're really interested in lately is working with marine microorganisms, usually bacteria, because they're a little easier to engage than the microeukaryotes, that are new, things that we've isolated that we know interact perhaps with diatoms or in some interesting way, and developing genetic systems for them, and then finding out how we can use these new tools to understand how they interact with their environment, how they interact with each other, and ultimately as a platform for new biotechnologies. So onto some science. This is like I said, the second research group that I've started. When you've got a new group and you get invited to give a talk, there's a panic mode because you're like, oh no, they're going to find out that my group hasn't done as much as we wanted to yet. But never, I guess I didn't have to panic because I can always fall back on some of my older research. So that's what I'm going to do today. And then follow up with some slides on what's going on down the hallway right now as we speak in our labs. So please let me introduce you or bring you to one of my former field sites, which is Hot Lake, Washington, in the US. We call it almost Canada because this is a field site located just a couple kilometers from the Canadian border, which causes problems often when we pull up in black SUVs and start unloading scientific equipment. Sometimes we would get checked out by helicopters. It's a very unique environment. We spent a lot of years there. Because it's a closed basin heliothermal lake. So we call it Hot Lake for a reason. It's quite warm. However, it's not geothermal. It's not a hot spring. And so the way it works is the sunlight heats it up, and because it's hypersaline, it's such a high concentration of magnesium sulfate salts in the bottom, it also has a high heat capacity, so it traps a lot of heat. But it's also stratified, so those upper layers that are less saline kind of act as an insulation barrier. So in the winter, this lake will actually get a really thin layer of amorphous ice across the top of it. But if you punch a hole in it and reach down, you can feel that the lake is still quite warm underneath the ice. So it's still trapping that sunlight and staying warm because of how much salt is loaded into the system. It's also a really extreme environment for life. It's warm, it's extremely salty. And you can see in this image that I'm showing you that it has, around the edges of this lake, there's this magnificent benthic mat ecosystem. So in the winter, it degrades. And as soon as the ice comes off, it builds back up and grows into this pustulent, beautiful film. I'll show you some pictures in a minute. can be up to a couple centimeters thick. So it's extremely productive microbial ecosystem. And like I said, unique. So we spent a lot of years as a larger group studying this ecosystem, also some hot spring ecosystems as part of a program to better understand how microbes interact in unique environments and perhaps maybe even discover some things. So I'm going to talk about two studies today that I was highly interested in, but if you were interested in Hot Lake or Hypersaline ecosystems related to Hot Lake, you could punch it into Google Scholar and you'd see several other studies from my friends and collaborators. But the studies I want to talk about today were driven by two initial hypotheses. And the first one was this idea that we expected productivity to correlate with species diversity. So you're looking at a picture of excised mat here, and this is extremely compact microbial ecosystem that is spatially structured. So it's easy to think, okay, sunlight is coming in from the top, that's where the productive zones are. But what is the species diversity associated with these different regions of this compact ecosystem? So when we started this, we drew upon an old ecological paradigm or the species energy theory. And this simply says that areas of ecosystems that have higher biological energy are those that can acquire the most biological energy and thus have the highest fitness, will also have increased diversity. So more productivity means that there's more niches that can be filled and you can have more species. And this is something that's been observed in macro ecosystems, in ocean upwellings or rainforest canopies, but to my knowledge, never before tested on microbial ecosystems in this way, definitely not on sub-millimeter spatial scales in compact ecosystems. The second hypothesis that we set out to do that year was based on community assembly. So I already told you that this lake ices over and this mat degrades. And then when the ice comes off, we have this benthic bloom and it matures up from essentially a thin biofilm all the way up to this pustulent looking mat that you're seeing in this picture. And based on preliminary data, and it's mostly metagenomic data, we knew that this thing is loaded with cyanobacteria. They're filamentous cyanobacteria. It's a cyanobacterial mat. Cyanobacteria are kings of this ecosystem. So we had this initial hypothesis that the first thing to arrive and start the colonization would be cyanobacteria. And then they would recruit other species, but sort of fill this niche of primary producers. And those that arrive first would have the priority effect and become the most dominant species, and thus giving way to what we were calling the cyanobacterial mat. So I'll just give the spoiler alert now that neither of these hypotheses turned out to be true, but the way we went about testing them taught us a lot about microbial ecology and this system in particular. So the first hypothesis that we went to test was the species energy theory, simply that the regions, the spatial regions within this map that have the highest productivity would also have the highest number of observable taxa. And so the way we went about that is we excised a mature microbial map from the lake and transferred it rapidly into this microcosm system, this flow through bioreactor or photobioreactor. And this gave us a couple of advantages. First, it's a very low shear stress system, so we can continuously replenish nutrients while still having direct access to the mat without having to reach down into the lake from the rubber rafts that we normally sampled it. But this also gave us the ability to sample it routinely for sequencing. So we would cut sections of mat and then we would take it to the microtome and cut them in 100 micron slices. And then we were able to sequence that and look at how community structure and diversity changes down the map in the context of measurements we were taking with microsensors, both for light and oxygen. But it also gave us the ability to do some stable isotope probing, which was really the major powerful tool we had for spatially assessing where productivity was in the system, at least with respect to autotrophy. So we would load the reactor, we'd pulse it with a stable isotope at night. So we had it on a 12-12 dark light cycle. We would load the stable isotope in as labeled bicarbonate at night, giving it time to diffuse into the mat. And then when the lights come on, we would give it another 12 hours to uptake the label. And then we could spatially profile that with a tool that my colleague developed called the Laser Ablation Isotope Ratio Mass Spectrometer, or the LAIRMS. And so we basically cut a cross-section of mat, such as I'm showing you in this picture, and you line up some crosshairs, and you fire a laser, which drills a 50-micron hole into the mat and ablates the material. And that material is then pulled up by a vacuum and combusted and sent to the IRMS so that we can get an exact spatial location of the isotope ratio. So it's a really powerful tool. We also really like this picture. We show it a lot. We call it the holy biofilm. So this is the sort of the characterization data that we got from that experiment. In panel A, you're looking at the simple stacked bar plot of 16S ribosomal RNA barcode sequences. I have to admit, I really like graphs. I don't get a lot out of them. But when you just assess it by looking at the most common taxa, you can see that it's really abundant, and cyanobacteria are what you would expect to be photosynthetic organisms. And also there's pretty consistent gradients down the system when you move from top to bottom. In panel C, you're looking at the spectral irradiance profiles. So we do this with a fiber optic microsensor with a 10 micron diameter. We probe lots of places and take averages. And you can see that light is rapidly attenuated right at the surface of the mat, but also those wavelengths associated with maximum absorbance of chlorophyll A and also phycobilisomes, a characteristic photopigment for cyanobacteria, are basically the first to be attenuated. So if you jump down to panel E here, this is just the scalar irradiance. So it's all wavelengths of light between 400 and 700 nanometers integrated. It's additional validation that this light is attenuated in the first top third or 25% of this map, and that's where you'd expect to see photosynthesis, which is in fact what we're seeing, and you see some evidence of that when you look at panel D, which is a dissolved oxygen profile that we get from a 25 micron oxygen sensing Clark type microelectrode. So you can see that this thing, there's a really high level of dissolved oxygen. There's so much photosynthesis happening. In fact, it's exporting O2 out into the overlaying environment. And you can see how it makes this hump shape. So we can see the position where there's this convex curve shows that photosynthesis is strong enough, it's outweighing respiration. And then when we get down past that maximum, we have the convex shape. And that's where respiration is beginning to outweigh photosynthesis. And then in panel F, I find this really interesting. It's something we didn't follow up on enough, but for me, it's kind of exciting. We got some measurements of porosity, just to get an idea of what the physical structure of the mat was as we went from top to bottom. You can see that the porosity is very low near the surface of the mat, and it's relatively high at the bottom of the mat. And I also provide this picture of it, and you can kind of see it when you play with this mat. It's a very rubbery structure. And the top is very gelatinous, and you can see it's probably this EPS bound biofilm where the things are probably mass transfer limited, and diffusion is the major and the dominant transport process. But down in the bottom, you can see those kind of white crystals, those white crusts. So there's a lot of carbonate mineral precipitation in this system. And we identified these at one point to be calcite, or at least some of them to be calcite. And it creates this more porous structure where nutrients and water can be transported across, or even microbes. So the physical structure changes a lot depending on whether or not you're in this photosynthetic productive zone or down in the bottom mineral layer. But the reason we did this study was to ask if, where in the mat, what is the most productive zone in the mat, and how does that correlate with productivity? So here's the measurements of productivity that we took. Panel A is from the stable isotope analyses, and we're able to estimate the percent of carbon replaced over 24 hours as it's been exposed to the 13C labeled bicarbonate or inorganic carbon. It's really no surprise to see that the most productive area of the mat in terms of autotrophy is right at the surface, because we know that's where light is available to drive photosynthesis. And panel B is, I guess, more of the same, but this is a measurement of gross oxygenic photosynthesis. So this is the rate of oxygen production in terms of micromoles of oxygen per second in the absence of respiration. And again, we're seeing that the major energy acquisition, at least in terms of water splitting, is happening right at the surface of the map. And that's what you'd expect to see. But I think the major punchline comes when we look at that in the context of alpha diversity. So here in panel A, you're looking at a profile down this map because we cut it in microsections and then sequence. And the total richness or total observed number of operational taxonomic units, or taxa, if you will. And what's interesting and what we didn't expect to see is the very surface of the map, indicated by the lighter colors, is actually one of the regions of low species richness. So there's less taxa at the very surface of the map where all this productivity is happening as compared to some of the deeper regions. And so naturally, and as you can see, and you've seen already, that sets up a negative correlation. So when we correlate that against the gross rate of oxygenic photosynthesis, and keeping in mind we only do that for regions that had a measurement that were above zero, we see a very strong and clear negative correlation. But also when we correlate it against the rate of carbon uptake or percent carbon replacement. And we see the negative relationship, but we can also surmise that, okay, we have autotrophic organisms taking up CO2 or the label, and they could exude some labeled CO2 that can be taken up in lower layers of the map. Even though the correlation is slightly less strong, we're still seeing this negative relationship. So there is in fact a negative productivity diversity relationship, which tells us that our initial hypotheses that we just kind of led with, which is the species energy theory, is not really supported. At least not in the way we looked at it, of focusing on prokaryotes and their relationship to photoautotrophic productivity. But we also learned that the community structure and the activity of this system changes on very fine spatial scales, or micrometer scales, which is the limit of our resolution. And that the maximum amount of carbon and energy uptake in the system occurs obviously at the surface of the map, which is a region of low species richness and high evenness, indicating that this is a specialist zone. So there are members of the community there that are very good at surviving in that region, and there's probably or likely some competitive exclusion allowing them to hold that area of the ecosystem to themselves, and thereby supporting the rest of the ecosystem or doing the most primary productivity. So the second hypothesis that we tested that and during that same field cycle, because we really enjoyed being there, was based on this community assembly and succession dynamics. So we knew that the system was seasonal. When the ice comes off, this beautiful, or maybe not so beautiful if you don't like biofilms, mat accretes up, and it starts from a very thin biofilm and becomes this very thick gelatinous structure. And so we wanted to look at how that happens under the hypothesis that we thought cyanobacteria would colonize this system, exert some priority effects, and dominate. So we did this in a 79-day in situ incubation where we deployed hundreds of sterile acid-washed glass slides into a grid made into the lake. And then we sampled them throughout the season in relatively high replicates, so 20 replicates for each type of analysis. And the major analysis we did was, again, amplicon sequencing with 16S ribosomal RNA as well as 18S. So we're also at this point probing and looking for the microeukaryote fraction of the community. And then we also took those slides and incubated them with stable isotopes, both 13C labeled bicarbonate, but also 15N labeled N2 gas. I'm just going to skip over that today to try to drive the punchline home. But we'd use that to support different bioimaging analyses that I'll show you in a moment. So what did we see in terms of community dynamics? I think the most important thing to point out here is if you look at the 16S, or the bacterial component of the microbiome, during the colonization periods, the early periods, there's a high species richness. So there's a lot of different taxa that we could observe. And this maintained pretty consistent until a point where there's a sharp drop off. So after this biofilm, so cells colonize the surface, they grow into a biofilm, and then they grow into a mature mat-like structure. And by the time it gets into this mat-like structure, it has a much lower species richness. So there's less taxa involved or that can actually be detected. We saw something similar with 18S, but less extreme and not so statistically significant. But it also kind of jumps out when we look at the community composition. So this is panel B, where we're looking at the most common taxa. And you can see that during colonization, cyanobacteria are the most abundant. But by the end, when this thing matures into a mat, cyanobacteria, while they're still very abundant, have kind of given way to proteobacteria, or a taxonomic class that more representative of heterotrophs or chemotrophs. And in terms of the micro-eukaryotes, we see that straminophiles are the most dominant, and we know these are diatoms. I'm going to show you that in a moment. And they start off dominant, but by the end, they essentially they're the most dominant. It's basically the only thing we're seeing to a point. And so it really becomes we see a major change in what colonized the biofilm to what ended up and what was part of this larger, thicker, more structured mat-like ecosystem. So we did a lot of bioimaging. We did a lot of it with confocal because it's relatively high throughput. We took hundreds of these images, so I'm just showing you some, obviously just representatives here. But after day one, so we deployed one of these slides, we came back and we took them just to see what has attached right away. We saw exactly what we expected to see, which is indicated by this red filamentous structure. So the blue is the nucleic acid stain, and the red is the chlorophyll A autofluorescence. And what we're seeing is the filamentous cyanobacteria that we thought dominated this map. But by halfway through, these slides are starting to grow a very thick, visible biofilm. And we're seeing something a little different, at least in terms of the chlorophyll A autofluorescence, in that the fluorescence we're seeing is bound in what are clearly these chloroplast-like structures. And if you look around them, you can start to see the outline of a very large pennant diatom. And we're seeing a lot less of these cyanobacteria, or at least chlorophyll around what would be these cyanobacteria. And by day 79, it's grown into this more mature mat-like structure, and it's essentially infested by these chloroplast structures and diatoms. However, it's a slightly different diatom morphology. It's still a pennant diatom, but it's a much smaller cell. So we're seeing three distinct stages in here where we go from, in terms of primary producers or even qualitative evidence, cyanobacteria to large diatoms to total infestation of smaller diatoms. We did a lot of nanosims on this, both with 15N labeled N2 as well as 13C labeled bicarbonate or inorganic carbon. And there's a lot of analyses behind this. And right now I'm just going to use it in a very cheap way of kind of driving the same point home that early on we're seeing activity around morphology structures that look more like these filamentous cyanobacteria. But later on, most of the activity we're seeing are in these diatom clusters, until the point at the end of the community where it's essentially infested with diatoms. We learned a lot about nanosims through this study, and that it's obviously not a great high throughput tool, but also diatoms are hard to work with nanosims because it's difficult to use the beam to get through the silicon frustules. So you never really know what's going on behind that, if you're not seeing activity or if the activity is just hidden from you. But when we go into the sequence of the analyses and start looking at the ecology of the system, or what we can learn about the ecology system from the molecular analyses, we can do something relatively simple to measure or estimate the successful recruitment of new species to the community. And you do that simply by saying that the fraction of previously observed taxa is equal to the fraction of previously unobserved taxa, which are assumed to be the sum of everything that you've already observed, divided by the sum of everything you're observing at that point. So if you use this formula and you look at your first data point, obviously everything's new. So you have everything is a founder species or a colonizer. But you can see at this point, by this curve on the left, that by the end of this experiment, the successful recruitment of new species is basically stabilized, and we're really not seeing new taxa in the system. So this means that the mat, after it's matured, or the growth of the biofilm, has either sampled all of the taxa that it could from around its ecosystem, or it set itself up in such a way that there's so much competitive exclusion that there can no longer be successful integration into the system. But for me, I think the most powerful analysis comes from looking at the beta diversity of this system. So here you're looking at the pairwise variance for every sample compared to every other sample with respect to community composition. And then we use the beta Jacquard metric for this, which simply generates a symmetric distance matrix. But the powerful thing about the beta Jacquard is it can be decomposed to be the sum of the variance observed due to the loss of species, or a term called nestedness. And this is, I'm showing this in this parameter beta JNE, or beta Jacquard nestedness, plus the variance attributed to the turnover of species. So this is either the loss of species and replacement by another, or drop in abundance by one group, taxonomic group, followed by gain in abundance by another. And what we've observed here is that turnover is by far the major contributor to the pairwise variance that we're seeing between every sample. So this becomes essentially the punchline of the study, is that turnover is the ecological process that is responsible, that is influencing the succession dynamics that we're observing as it goes from basically a colonizing cell to a thicker biofilm all the way up to this mature microbial mat that has all these wonderful spatial gradients that I showed you earlier. So we dug a little deeper into that, into an analysis that we called the founder species analysis. And we simply look at the taxonomic groups at the phylo level that are in highest abundance over a threshold, which in this case was 5% of the community at colonization. We said, okay, those are the colonizers. And then those that are in high abundance post-colonization at day 79 at the final are considered not founder species. And then we tracked which specific taxa all the way down as far as we could go with taxonomic analyses from 16S or 18S classification. Which ones are responsible, and do they group as an original group? Are they founder species, or are they something that came that bloomed up in the system later? And so what we're seeing is, again, cyanobacteria get there first, but it's largely attributed to a single taxa. The Firmidium, and we knew about this. We knew it was there. We'd seen it. We'd worked with this species. We didn't have it in isolation, but we had its metagenomic assembled genome in other data sets. So this is what we expected to be the queen bee of the entire system. But in fact, even though it was the major colonizing species, it gave way in its dominance to other taxa, and specifically this rhodobacter, which is a taxa more representative of chemoautotrophy, or certainly not a cyanobacteria, a photoautotroph. So even more powerful than that is what we observed from the micro-eukaryotes. And so I already showed you that diatoms show up at some point, and then just that, at least in the images, that's all we can see. There's these pennant diatoms, but it's pretty extreme when we look at it here. That of the founder species, we have different groups, and some of them, many of them are diatoms. But then by the end, it's basically a single winner that takes all, and it's this Nichia taxa. And I think that the fun thing about diatoms, whether I agree with it or not, is it's closer to plant science, and that you can still look at the morphology of the cell and make some conclusions about the taxon. I think that's changing in the world of sequence as many genomes as possible. But when we look at this in the literature, this Nichia cell, it very much is representative of this the smaller pennant diatom that we were seeing infest the system later. And the abundance of it is striking. At least in this case, it dominates everything. So what did we learn? We learned that turnover actually defines the succession dynamics in the system, and that it strongly outweighs any of the notions that we went into the experiment with. The priority effects would be very important. The most abundant taxa, once the system went from a biofilm to a more mature mat, were in fact not founder species. But we did find out who the founder species were, and when we looked at the ones in at least in highest abundance during the initial colonization period, we can attribute that to two different cyanobacteria, one taxa that is representative of a chemoheterotroph, and two different diatoms. New species were rapidly recruited to this community, so it starts off as a jungle, a really very diverse system. But essentially, there were no new colonizers or no successful recruitment of new species after 56 days or 75% of the way through the succession experiment. And the diatoms take over the system about midway through, and there's two distinct stages of that, at least from a morphology perspective. There's these large diatoms that start to arrive, and then they give way to infestation of these smaller diatoms, or these Nietzsche cells, that were very apparent both from the microscopy as well as the molecular analyses. So Hot Lake was a lot of fun. I spent a lot of years there. I still think about it a lot. We had rattlesnakes, we had bears, we had sampling grounds, we had great times there. But now I live in northern Norway, and we're rebuilding a lot of things. And so I want to just take a couple of slides to show you about what's coming next. Probably from starting my work in Hot Lake, I became very interested in diatoms and how they interact in larger microbial ecosystems. And so we're very much focused on that right now. We have a really large project working with diatoms for biotechnology and carbon capture and utilization, where we're just simply growing as much diatom biomass as possible on industrial CO2, and then harvesting that and trying to use it as additives in aquaculture feed. But from an ecology perspective, I think that these are amazing animals. They structure entire ecosystems around them, at least microbial ecosystems. And so what I'm showing you here is a picture of a pennant diatom with bacteria attached to it, and we can define this as the phycosphere. And so one can assume that these bacteria are dependent on organic carbon produced from the diatom or photosynthate. And so right now, actually, literally right now down the hallway, we are working with diatoms that we have grown up to be azenic. So we harvested them from the local fjords, and also some from very far north on the ice edge. We treat them with antibiotics so that they don't have any bacteria, or we can try to keep them clean. But at the same time, we take seawater and filter away the diatoms where we have the bacteria component. And then we can recombine those and look at how the diatom, which bacteria it's selecting for, and how it's restructuring the environment around it. And the tool that we're using a few different tools, but the one I think we're most excited about using right now is DNA-SIP. So we feed it 13C labeled bicarbonate, and we're trying to pick that up in the heavy DNA of the bacteria. And this is working pretty well. We definitely are able to separate into heavy bands, and we're going through the process of amplicon sequencing on this. So again, just 16S. But I think what we're really excited about, and also learning from people who have done it before, is amplified metagenomes. So we can really go after functions, who's interacting with the diatoms and what is their genomic potential. And then the other thing that I'm really excited about that we're working on right now is doing a lot of synthetic biology and cold adapted marine microorganisms, also some soil microorganisms that haven't historically been thought of as very genetically tractable microbes. And so we spend a lot of time trying to build things like toggle switches and repressolators and CRISPR interference devices into them just to see if we can. It's kind of a hobbyist way of doing biology. But we're now arriving at this point where we're starting to get some of these organisms that we know interact with diatoms, and we're able to engineer them. So we have toggle switches built into a few of them, and we're gearing up to start running these experiments where we can grow them in combination with the diatoms. So you can think of the diatom as an engine. It's fixing the carbon, it's gathering the energy, and it's supplying that to the bacteria that is the controllable modular device that we can actually do things with. So we're looking at how different bacteria influence the performance of genetically engineered devices, but also looking at how their relationship to a diatom or the primary producer influences the stability of these devices and also their ability to host them and be programmable. So I think we'll get some new tools out of this that allow us to interrogate microbial interactions and perhaps even program them. And in the long run, this will probably lay the groundwork for some new biotechnologies in the area of carbon capture and utilization. So I'd like to thank people. Nothing happens in a vacuum. The research that I showed you, a lot of it happened just kind of right before I moved to Tromsø here. And so I'd like to thank a lot of my former colleagues at PNNL, specifically my colleague James Moran, or Jim Moran, who's an expert in stable isotope analyses. And he invented and built the laser ablation isotope ratio mass spec. We got a lot of mileage out of that. But I also like to give a shout out to my current postdoc, Hannah Schweitzer, who's doing a lot with these diatom-bacterial interactions, and my current PhD student, Dennis Chan, who is a whiz in cloning and transforming non-model microbes. So we're getting a lot of devices built into things, just based on his skill and the luck factor. And also everybody all the different institutions who donate resource in terms of funding and infrastructure. So with that, I would like to thank you for sitting through. I should give you this serene picture of one of our field sites that's just down the fjord from where I'm sitting right now, and it'll open it up for questions and discussions.
Thank you very much, Hans, for a fantastic talk and for giving so detailed insights into those communities in biofilm, and also to provide a perspective of what you are working on right now, which is super cool. And yeah, I would like to open up the floor to any of you who have questions. And I see Kylie, please go ahead.
Hey, I really enjoyed your talk. That was great. I was curious because you looked at the microbe accession, but I was curious how that tied into the different layers you saw in the map. Like, does one layer appear first and that's the microbial community you're seeing and then it changes?
That's a good question. The experiment wasn't designed to tackle succession across the one-dimensional gradient, because as you can imagine, the number of samples starts to explode. And we have no idea like what arrives first. I mean, we know that their phototrophs are early colonizers, and if they're going to be fit, they need to be at the top of the map. But I think one of the things we learned through that experiment, is how does a biofilm and benthic ecosystem form? Does it accrete from the bottom, or is there sedimentation that makes it, that creates it? Sedimentation or things coming in from the top and attaching, or is it a combination of both, which I think it is, and which one's more important? So we didn't do it on a spatially resolved location, or at least not the succession, but we should, and I want to. That would be an exciting thing to do.
Thank you.
Any other questions for Hans from the round? I also check the chat. Well, maybe oh, yeah.
Hi, Hans, thanks. Cornelius Gross from EMBL in Italy. It seems like spatial dimensions are really important for you, one-dimensional, two-dimensional, three-dimensional. So I imagine there's, I just want to hear more on the technological side, imaging methods that you're working on or you see on the horizon that are going to be really important for pinpointing processes. exchanges between these organisms at much higher resolution somehow. Do you see that as an important point? And what do you see on the horizon? What's technologically?
Yeah, absolutely. I think that's probably why I'm here today, is because I met Yannick Schwab, and he does some amazing imaging, and I'm obviously thrilled to see what a lot of you guys are doing with subcellular imaging and connecting that back to like the transcriptome or different molecular signatures. I would love to get to do that on some of the systems we work with. I think we're a long ways from that. I think that what could happen and what could be really exciting is using things like activity-based probes, or even just like probes that show translationally active bacteria such as BONCAT, and looking at some of these aggregates such as like bacteria attached to a diatom, and looking at using that to identify where certain cells are active, and then later disaggregating them, running them through flow cytometry or some flow cytometry imaging, and then pushing that through single cell sequencing. So what I'd really like to look at is the specific genomes of specific cells in an aggregate, and understand, you know, what can they do in terms of genomic potential, but how are they assembled? I think for us, that's probably using a combination of activity-based labeling, flow cytometry imaging, and single cell genomics. It's probably it's probably on the horizon in the next year or two, or something we're talking about and trying to build collaborations around being realistic.
So if I got that right, if I can just follow up. So you're thinking of genomes, though. You think there's different species that are going to be in different positions. But I mean, if you think of multicellular organisms, you have epigenetics, and the ones that are close just have a different gene expression pattern, same species, than if they're far away. So you think it's all genetics, or don't you have to look at sensors and gene expression patterns?
Yeah, I would love to look at gene expression patterns in a spatial resolved way, and I know a lot of you guys do that. I guess when you're sitting in a smaller university like Tromsø, especially after you just moved from a big institution, you're always looking at, okay, what's the first step, and how do I start to peel this onion back? But of course, we want to know everything about them. So,
Thanks.
And I also do have a question, if I may. I found it very interesting what you showed at the end, that you have diatoms that interact in a somewhat specific way with bacteria. And so you said that, well, you do have the engine, the diatoms that produce carbon that then the bacteria use. So my first question there is, do you have any idea what actually the carbon source is that the bacteria go after from the diatoms? And then is there something in for the diatoms as well? What do the bacteria give back, or is there nothing? So is it not like, we shouldn't think of a symbiotic interaction between the two?
I think that there's a lot of evidence of two-way exchange, mutualism, and commensalism. Obviously, some of them are even parasites. In terms of diatom-bacterial interactions, I'm relatively new to it, but for the last few years, I've been looking at cyanobacteria heterotroph interactions, and there's all sorts of two-way exchanges. Of course, this is a prokaryote system. But where we saw probably the largest amount of two-way exchanges that resulted in what we thought was mutualism were through amino acids, especially those that are metabolic costly, or the nitrogen-carrying amino acids. That's the big question for us in diatoms. My postdoc is really interested in following up on quorum sensing. There's some evidence that there are some quorum sensing abilities that cross from the prokaryote world over to the micro-eukaryote world. She's really interested in that. I think that I'm interested in knowing about hormones. So are there hormone-like compounds being produced by the prokaryotes that are influencing the eukaryotes? And I expect what we're going to see is a jungle, that like anything you can imagine, really, there will be some evidence for. So I think the real challenge we have is being specific about it. this compound exchanged to that from this species to that species, when and how.
Very interesting, and it seems with your germ-free diatoms, you have a very cool system to actually do it systematically and bottom up. So, very exciting work.
Yeah, thanks. I should be clear, like we keep azenic diatoms, but it depends on which week it is or which day of the week it is. It's a tremendous amount of work to keep them clean.
Yeah. Are there any other questions from the round for Hans? Please raise your hand. If this is not the case, then well, let's thank Hans again for a great presentation. Thanks a lot for joining us today, and well, thanks all of you for joining the seminar.